This is the first part of 3 of the talk Fungal Bioluminescence, Mechanism and Ecological Function about the work of the Laboratório of Fungal Bioluminescence at the Chemistry Institute of the University of São Paulo, coordinated by Professor Cassius Vinicius Devani. Narration by Imran Virmal and edition by Henrique Domingos from IPBio, the Biodiversity Research Institute, or Instituto de Pesquisas da Biodiversidade, an organization that collaborates with LBF. Our work has been featured on the cover of scientific journals, magazines, newspapers, TV, radio, and even documentaries. The interaction with journalists is very important to promote our work, and to attract students and financial support from national and international agencies and companies. One of the species we discovered, Mycena lucentipes, appeared in 2018 in a commemorative stamp of the US Postal Service. This talk is divided in 3 parts. This is the first part, and we'll introduce you to basic concepts of fungi and bioluminescence and show you beautiful images of our fieldwork and mushrooms. In part 2, we'll talk about the chemical and biochemical mechanisms involved in fungal bioluminescence and biotech applications. And finally, in part 3, we'll talk about the hypothesis for an ecological function of bioluminescence. All fungi that emit light are Basidiomycetes, a phylum of fungi that produce mushrooms, live in soil, and feed on cellulose. Their life cycle begins with a spore, which produces a specialized filamentous cell known as hyphae. Hyphae of different fungi, but the same species can mate, yielding a network called mycelium, which is the fungal organism. In the right conditions of pH, humidity, temperature, light, and nutrients, the mycelium produces a young mushroom that is transformed into a mature mushroom. The mushroom is divided into stipe, the stem, and pileus, the cap. Under the pileus are lamellae, gills, where the spores are produced. Spores can be taken by the wind and reinitiate the life cycle. Diverse insects, like flies, can be attracted by either the smell or the bioluminescence of the mushroom. Upon laying or feeding on mushrooms, insects can be covered by fungal spores and transport them far, far away, then helping the fungus to spread the spores. Currently, there are 100,000 fungi described in the literature, but the estimation is that there are between 1 and 3 million species. 21% of fungi described are Basidiomycetes. 9% of them are Agaricales, in which occur all of the 105 bioluminescent known species. Bioluminescent fungi are present in the whole world, from Pacific Islands passing by the Americas, Africa, Europe, Asia, to Japan. A third of the total species can be found in the Americas. They belong to 4 different lineages: Mycenoid, Lucentipes that only exist in Brazil, Armillaria, and Omphalotus. We are responsible for the discovery of almost 20% of the described species. Bioluminescence can be described as a biochemical emission of visible light by living organisms, such as fungi, fireflies, and deep-sea fishes. It's a chemical reaction of oxidation, but instead of releasing heat, it releases energy in the form of light. In order to occur, all bioluminescent systems require an oxidant such as molecular oxygen or hydrogen peroxide. In fact, there is no bioluminescence without oxygen. a substrate, generically called luciferin, a catalyst, generically called luciferase, or a photoprotein, depending on the turnover of the protein. Some systems require cofactors, such as NADH, NADPH, or ATP. Bacterial systems require NADH or NADPH, and fireflies require ATP. Cations, as magnesium for fireflies or calcium for jellyfish. Auxiliary proteins, as reductases, or the green fluorescent protein GFP. Regarding the type, bioluminescence can be direct or indirect. Direct bioluminescence takes place in fireflies and fungi. The luciferin is oxidized by the luciferase in the presence of molecular oxygen, yielding oxyluciferin, the oxidized luciferin, in the singlet excited state, which emits light upon its return to the ground state. Indirect bioluminescence occurs in jellyfish, for example. In this case, the emitter is not oxyluciferin, but the auxiliary protein GFP, which is excited by oxyluciferin following bioluminescence resonance energy transfer, or BRET shortly speaking. Until 2020, there are 10 described luciferins, 9 of them can be seen here. It can be promptly noticed that their structures are very different. As previously said, luciferin and luciferase are generic terms and should be used accompanied by the name of the organism that they refer to. Our work began in 2001 at the Rio Ribeira River State Park, Petar, or the Parque Estadual Turístico do Alto Ribeira, in Iporanga, São Paulo, Brazil. In the beginning it was tough. We camped in the middle of the Atlantic forest and brought the equipment on loaded mules. From the beginning, we were able to discover and identify 20 of the 105 species described in the world. Most of them belong to the mycenoid lineage. The Lucentipes lineage, as already mentioned, only exists in Brazil. They probably belong to a new genus of bioluminescent fungi. In 2008, we began a fruitful collaboration with IPBio. Ever since, our work has gained momentum, and we are able to speed up the process of finding new species. During the day, you probably won't notice a common dead leaf, but at night, with the headlights turned off, it's not a mere leaf, it glows, showing us that the place is full of bioluminescent mycelium and probably mushrooms as well. A small log. It's not just a log. It can also indicate the presence of bioluminescent mushrooms. We collect during the new moon mostly, or when it is cloudy, or when the moon is not up in the sky, because the light of the moon reflects everywhere and we cannot see the mushrooms. This is Neonotobanus Gardineri, also called flor de coco in Piauí and Goiás, from where it was first described by George Gardner in 1840. It's one of the brightest and biggest bioluminescent mushrooms ever found. It occurs only in Brazil, and it was very important in unraveling the mechanism of fungal bioluminescence, together with its Vietnamese relative Neonotobanus nambi. This is Mycena lucentipes, described in 2007 and the cover of the Mycologia journal. Its name means luminescent stipe. That's why it's one of the representatives of the Lucentipes lineage, along with Geronema verdelucens. Here's another photo with a small log with a bunch of Mycena lucentipes, took in December 8th, 2002, Jim Morrison's birthday. This beautiful composition was the picture that appeared on the cover of Mycologia journal in 2007. This is Geronima viridescens, the first species we found in Petar, back in 2001. Its colors vary from red to beige. This species was also very important in the beginning of the project, as we developed a fungal-based ecotoxicological luminescent assay with its mycelium. The assay can be used to evaluate the toxicity of inorganic and organic chemicals to the Basidiomycetes fungi. Mycena asterina was also described in 2007. It seems big in this macro photo of David Lutzwiger, but it's only 1 inch tall. It received its name because it only emits light in its cap. The place it was first found in the forest litter had hundreds of them, emitting light like little stars in the sky. Hence its name in Latin, which means stars, aster. Mycena oculus nymphae, or Mycena fairies, was described in 2016 by Desjardins, Perry, and Steveni. It's a very delicate mushroom and was described with 3 more species found in Petar: Mycena globulispora, Mycena deformis, and Rhizonomycena petarensis. Mycena luxiterna also appeared on the cover of an issue of Mycologia in 2010. Its name means eternal light in Latin. It emits light only on the stipe, as Mycena lucentipes, and has a jelly-like substance on it. In 2011, it was chosen as one of the top 10 new species. This means it was one of the 10 most interesting species described among circa 10,000 others described the year before. This is a time-lapse of Mycena lucentipes mushrooms growing. It took about 12 hours to obtain the photos, as mushrooms grow slowly. Meanwhile, they can be eaten by animals or decomposed by bacteria and other fungi. This is another photo of Neonotobanus Gardineri growing on the base of a babaçu palm in Altos, Piauí state, Brazil. There are 2 spiders near the mushrooms. One of them is quite obvious. The other one is on the top of the mushroom on the left. Can you see it? Spiders use the light of the mushrooms to hunt insects and other animals. We're going to return to this subject in the third and last part of the talk. Thanks for watching.